Hello, good evening, everyone. This is chapter the cytoplasm for histology, and we studied about the introduction about the what is a cell, what is the extracellular matrix, the cytoplasm. We also did cell differentiation. You can check this video on the live section. Uh, let's get into the plasma membrane. Okay, the plasma we'll study along. I'll read. I will try to explain, and you just have to focus, listen, grab a pen and paper, write down important notes in your language, in the language I tell. Do what's convenient for you. Let's get into it. The plasma membrane. The plasma membrane that envelops every eukaryotic cell consists of what? Consists of phospholipids, consists of cholesterol, consists of proteins with oligosaccharides chains covalently linked to many of the phospholipids and proteins. This limiting membrane functions as a selective barrier regulating the passage of materials in and out of the cell and facilitating the transport of specific molecules. One important role of the cell membrane is to keep constant the ion content of cytoplasm, which differs from that of the extracellular fluid. So, plasma membrane is basically a double membrane structure which surrounds the cell. What does it consist? Four important things: phospholipids, cholesterol, proteins, and oligosaccharide chains. Okay, membrane proteins also perform a number of specific. We also read here what is the function. The function is to regulating the passage of materials from in and out of the cell. That is important. Let's highlight this passage of material. Okay. One important role of the cell membrane is to keep constant the ion content. Even this is a role. Keep constant the ion content of cytoplasm. Also perform a number of specific recognition and signaling function, playing a key role in interactions of cell with its and within its environment. With its environment. Okay. Although the plasma membrane defines Although the plasma membrane defines the outer limit of the cell, a continuum exists between the inner of the cell and the extracellular macromolecules. Certain plasma proteins, the integrins, are linked to both the cytoplasm and the extracellular matrix component and allow continuous exchange of influences in both direction. Exchange of influences both direction between the cytoplasm and the material of the extracellular matrix. So as we saw the function of the cell membrane that it differentiates the cell cytoplasm and the extracellular matrix, but there is a continuum always maintained through what? This continuum between the cytoplasm and the extracellular matrix. It's maintained through what? It's through a protein called integrins. Integrins are basically linked to the both cytoskeleton. Cytoskeleton is basically in the cytoplasm and the extracellular matrix, which is outside of the cell. And this is the continuum that is developed, that always exists. Okay, membranes range from 7.5 to 10 nanometer in thickness. In histology, we are always going to keep highlighted the matrix, the important diameters, the important thicknesses of each specimen. Then I think some universities really go about asking this type of questions. Some universities go really logical questions. It really depends, but we have to have all our options and everything prepared for the exam. USMLE doesn't go for this type of questions, but this type of data is important for your university exams. Okay, membranes range from 7.5 to 10 nanometer in thickness and consistently are visible only in the electron microscope. The line between the adjacent cells, sometimes seen faintly with the light microscope, consists of plasma membrane proteins plus extracellular matrix, which together can reach a dimension visible by light microscope. So in the plasma membrane, because it's in nanometers, we cannot see it with the bright field microscope. We need an electron microscope. But sometimes some proteins which are having a bigger thickness can be seen through a light field microscope. Membrane phospholipids are amphipathic, consisting of two non-polar long chain fatty acids linked to a larger polar hydrophilic head that bears a phosphate group. So phospholipids is lipids with a phosphate group. So they are divided into two parts: the non-polar chain, a polar head and a non-polar tail. Polar head, as it says, polar, it's hydrophilic, means water attracting, and hydrophobic, non-polar, means water repelling. That's why this nature, because of this type of arrangement, we call this membrane phospholipids are amphipathic. Amphipathic, sorry. Phospholipids are most stable when organized into double layer, with the hydrophobic fatty acid chains located in the middle region away from the water and hydrophilic polar head group contacting the water. Molecules of cholesterol, a sterol lipid, insert at varying densities among the closely packed phospholipid fatty acid, restricting their movements and modulating the fluidity of all membrane components. Okay. The phospholipid is, I think this part is important. The molecules of cholesterol, sterol lipid, insert at varying densities among the closely packed phospholipid fatty acid, restricting their movement and modulating the fluidity of the membrane components. So, what is the role of cholesterol? What does it do? It moderates the fluidity. Mod- Restricting the movement and moderating fluidity is the role of cholesterol and sterol lipid. It just keeps the phospholipids packed. For example, the well-studied membrane of red blood cells, uh, phosphatidylcholine and sphingomyelin are more abundant in the outer half, while the phosphatidylserine and phosphatidylethanolamine are more concentrated in the inner layer. This part is important. Phosphatidylserine and phosphatidylethanolamine are in the inner layer of the cell membrane. These are the phospholipids that we have as an arrangement as a bilayer, and phosphatidylcholine and sphingomyelin are on the outside of the cell membrane. Okay, so sorry. Yes, so okay. Some of the outer layer lipids known as glycolipids include oligosaccharide chains that extend outward from the cell surface and contribute to a delicate cell surface coating called glycocalyx. So some of the outer layer lipids known as glycolipids. So these glycolipids just protrude out of the cell membrane outside of the cell, and these type of glycolipids collectively give rise to a structure, a kind of coating, which is called glycocalyx. What is glycocalyx? Consists of glycolipids. With the transmembrane mission, the transmission electron microscope, the cell membrane as well as all cytoplasmic membranes may exhibit a trilamellar appearance after fixation in osmium tetroxide. As we know, in the previous chapter, we read about the how a specimen is prepared. We saw with that we use fixatives, and osmium tetroxide is the most common fixative. But when uh, osmium tetroxide is used as fixative in case of cell membrane, it gives a trilamellar appearance. How does it give a trilamellar appearance? Let's check. So osmium binds the polar head of the phospholipid and the oligosaccharide chain, producing two dark outer lines that enclose the light band of osmium free fatty acids. So I think the we have a polar head, a non-polar chain, then again a non-polar chain and a non-polar head. So osmium, I think, binds to the polar heads of the phospholipid, and we have a chain arrangement here, so it gets a trilamellar arrangement. Proteins are major constituent of membranes. Almost 50% by weight of the plasma membrane is protein. Okay, important. Integral proteins are incorporated directly within the lipid bilayers, whereas peripheral proteins are bound to one of the two membrane surfaces, particularly on the cytoplasmic side. Look, guys, we have studied this chapter is going to be very, very uh, correlated with the knowledge we had studied already in the pre-med. Cell membrane was widely discussed in pre-med chapters as well. But let's take a look and let's complete what we have here. Integral proteins and peripheral proteins are the most proteins that are found in the plasma membrane. As the name suggests, integral protein is the protein that is incorporated throughout the bilayer, whereas peripheral protein is only present at one of its layers. Okay, so peripheral protein can be extracted from the cell membrane with salt solution, whereas integral proteins can be extracted only by using detergents to disrupt the lipid, to disrupt the lipids. Okay, so I think this part is something important. If you want to extract this type of proteins from the cell membrane, what we can use, the reaction with salt solution will help the peripheral protein to extract, and detergents will use integral proteins to extract. Okay, the polypeptide chains of many integral proteins span the membrane from one side to the other several times and accordingly called multipass proteins. Okay, the polypeptide chains of the many integral proteins. Guys, we have to remember that integral proteins can only be multipass proteins. And multipass proteins from one side to the other several times according to. So basically, integral proteins span the membrane, and that's why they are from one side to another, and that's why they are called multipass protein. That's it. Integration of the proteins within the lipid bilayer is mainly the result of hydrophobic interactions between the lipids and non-polar amino acids of the protein. Integration of the protein within the lipid bilayer is mainly result of hydrophobic interaction. Yeah, understood. Like, we need hydrophobic interaction. I think that is why the concept of amphipathic plasma membrane makes sense for to hold the proteins in their place. So I think that's why the interaction of the protein within the lipid bilayer is mainly the result of hydrophobic interactions between the lipid and non-polar amino acids of the protein. Let's also have this important mark. Hydrophobic interactions. We have to remember these are the reactions that is a result for the place of these proteins. This is a reaction between the lipids and the non-polar amino acids. Freeze fracture. Electron microscope study of membrane show that part of many integral proteins protrude from both the outer or the inner surface. So freeze fracture is a type of technique which is it's preparation of a specimen with liquid nitrogen, and then we we freeze them with a liquid nitrogen, and we cut them in specific shape, and then observe it under an electron microscope. So that's why it's called freeze fracture electron microscope. But what's important here is that this type of study shows that parts of many integral protein protrude from both inner and outer surfaces, and that's why they are called integral proteins, because they are present throughout the plasma membrane. Like those of glycolipids, the carbohydrate moieties of glycoproteins project from the external surface of the plasma membrane and contribute to the glycocalyx. So, as we read about glycocalyx already, some glycolipids from the outer layer protrude outward and give a coating structure. So, as integral proteins are also observed protruding outside, they also contribute to the appearance of the glycocalyx. They are important component. They are important components of proteins acting as receptors, which participate in important interactions such as cell adhesion, cell recognition, and response to protein hormones. So this type of proteins that protrude outside uh, have an important role, and that role can be cell adhesion, cell recognition, response to the protein. And when a protein is into these three types of roles, it's called receptor. So what are receptors? Basically, they are integral proteins. And but we have to know one thing: not all proteins are receptors, but all receptors are proteins. Okay. As well as lipids, the distribution of the membrane polypeptide is different in two surfaces of cell membrane. Therefore, all membranes in the cell are asymmetric. Yeah, so we know like the distribution of the proteins, the polypeptides is different, and that's why it makes the membrane asymmetric. Somewhere it's integral protein, somewhere it's peripheral proteins, and whatsoever type of arrangement is not fixed, and that's why we have an asymmetric membrane. Okay. Studies with labeled membrane protein of cultured cells reveal that many such proteins are not bound rigidly in place and are able to move laterally. Such observations are well as data from biochemical electron microscope and other studies show the membrane protein comprises of a movable mosaic and fluid lipid bilayer, the well established fluid mosaic model. So the fluidity. Okay, so the movement of the proteins along the membrane is observed. And what kind of movement is this? It's a flipping movement, and this type of movement has been observed through electron microscope, and there are a lot of studies that prove this, and that's why this type of model of flipping proteins, and we have the lipid bilayer, turns out to be called as fluid mosaic model. Why the fluid word comes from the fluid nature of the lipid bilayer being a polar head, means hydrophilic, and we have mosaic word comes from this movement of proteins, making this name as fluid mosaic model for membrane structure. Unlike the lipids, however, lateral diffusion of many membrane proteins is often restricted by their cytoskeletal attachments. Moreover, in most epithelial cells, tight junctions between the cells also restrict lateral diffusion of unattached transmembrane proteins and outer layer lipids, producing different domains within the cell membrane. So we are going to study about different types of junctions in chapter 4. So let's check them out there. Membrane proteins that are component of membrane proteins that are component of large enzyme complexes are often usually less mobile, especially those involved in the transduction of signal from outside the cell. Okay, so here is an important thing that needs to be understood. So membrane protein, we read about types of proteins we have on our membrane, but some membrane proteins are components of large enzyme complexes. They are involved in a large enzyme complexes, which make them less mobile, and they are mostly the proteins that are responsible for transducing signals from outside the cell to inside the cell. Let's check about such protein complexes are located in specialized membranes. Membrane patches terms are, so this type of membrane patches where we have a protein has a complex with enzymes and it moves less, and this type of complex is responsible for transducing signal from outside the cell to inside the cell. So these are called lipid rafts. They're termed as lipid rafts. With higher concentration of cholesterol and saturated fatty acids, with reduced lipid fluidity. Okay, check. I need to check my mic, guys, for a second. Check, check. I'm not sure if everything is okay. Please give me a second. If there is anyone on the chat, please tell me if things are okay. Arrangement is not fixed, and that's why we have. Yes, yes, it's all okay. So, sorry, I was just checking if my voice is fine. No worries. All right. So we know now what is lipid rafts. They are. We also need to understand one important thing, that lipid rafts which also has high concentration of cholesterol and saturated fatty acids, which are, that's why, responsible to reduce lipid fluidity, making it less mobile. So this, together with the presence of scaffold proteins that maintain special relation between enzymes and signaling proteins, allow the protein assembled within lipid rafts to remain in close proximity and interact more efficiently. So we talked about these proteins that are involved with enzyme. They are called scaffold proteins. We need to know, remember what are scaffold proteins. They are proteins involved with the enzyme complexes, creating a lipid raft around what is lipid raft, where we have high concentration of cholesterol and fatty acids. These make the fluid, they reduce the lipid fluidity in the plasma membrane. Okay, let's now check about transmembrane proteins and membrane transport, how proteins facilitate the transport throughout the membrane. So transmembrane proteins, the plasma membrane is the site where materials are exchanged between the cells and its environment. Most small molecules cross the membrane by general mechanism shown schematically in figure 2.5 and explained as follows. Let's check figure 2.5. Oh, we have table 2.5 here. We have simple diffusion, we have transport through channels. We're going to study out channels in detail in later chapters, and we have carriers. Let's check what all this means. Okay, so we have diffusion. Diffusion transport small non-polar molecules directly throughout the lipid bilayer. Direct transport. Lipophilic, that is fat soluble molecules, diffuse through the membrane rapidly, water very slowly. So diffusion is basically the uh, material goes from higher concentration to lower concentration directly by diffusion process. This is called diffusion. And water, diffusion of water goes very slowly. Channels are multiplast proteins forming transmembrane pores through which ions or small molecules pass selectively. So channels, as we're going to study them much detail in later in much detail, but sorry, we will study junctions much details, but junctions are some type of channels only. So channels, they are multipass proteins. We remember in the last minute we read about multipass proteins. What type of proteins are multipass proteins? They are all integral protein, guys. The multipass proteins. Cell opens and close specific channels for Na plus, K plus, calcium 2 plus, and other ions in response to various physiological stimulus. So, as and when required, these channels are open for specifically mostly ions. Also, water molecule usually cross the plasma membrane through the channel proteins called aquaporins. So, as we saw that diffusion is the process through which water transports very slowly, but the efficient way for water transport is through the protein channels that are called aquaporins, specifically for water. Carriers are transmembrane proteins that bind small molecules and translocate them across membrane via conformational changes. So we are here, we know carriers, simple transmembrane protein that bind small molecules and just translocate them inside the membrane. Diffusion channels and carrier proteins operate passively, allowing movement substances across the membrane down a concentration gradient due to its kinetic energy, as I said, from higher concentration to lower concentration. In contrast, membrane pumps are enzyme engaged in active transport utilizing energy from the hydrolysis of ATP to move ions and other solute across membranes against often step concentration gradient. So these three methods we saw are all passive, do not require any kind of energy. There is an active pumps that is an active transport where sometimes things can be transported against the concentration gradient as well because they use ATP, and that's why we can go against the concentration gradient. These transport mechanisms are summarized with additional details in table 2.2. We'll go and read again table 2.2 in detail, but let's check what we have further here. Okay, let's go. Transport by vesicles. Vesicles, as we know from our basics, that vesicles are basically a vacuole, a circular material in the form of circular vacuole. And let's see how are these vacuole transported in and out of the cell. So macromolecules normally enter cells by being enclosed within a fold of plasma membrane. So following often after binding specific membrane receptors, which fuse and pinch off internally as spherical cytoplasmic vesicles in a general process known as endocytosis. So basically, endocytosis is this general process where plasma membrane folds and engulfs a specific type of vacuole, a vesicle, and this type of process is called endocytosis. Three major types of endocytosis are recognized and summarized in table 2.2 and figure 2.6. We will go and check them, but let's read the text first. Phagocytosis. The first type is phagocytosis. Phagocytosis in the lemon language is cell eating, is the ingestion of particles such as bacteria or dead cell remnants. Certain blood derived cells such as macrophages and neutrophils are specialized for this activity. So among the five blood cells, two important macrophages and neutrophils are responsible for the phagocytosis. When a bacterium becomes bound to surface of the neutrophil, it becomes surrounded by extensions of plasma lemma, that is plasma membrane, and cytoplasm, which project from the cell in process dependent on cytoskeletal changes. Fusion of membranous folds encloses the bacterium in an intercellular vacuole called a phagosome, which then merges with a lysosome for degradation of its content, as discussed later in this chapter. We will study what are phagosomes later, but fusion of the membrane fold enclosing the membrane bacterium in their intercellular vacuole is called basically phagosome. That structure. Pinocytosis is cell drinking. Involves smaller invaginations of cell membranes which fuse and entraps extracellular fluid and its dissolved contents. Okay. The resulting pinocytosis vesicle almost 80 nanometers in diameter, then pinch off inwardly from the cell surface and either fuse with the lysosome or move to the opposite cell surface where they fuse with the membrane and release their content outside. The later process, called transcystosis, accomplishes bulk transfer of dissolved substances across the cell. So, so many things here. Pinocytosis, cell drinking. Basically, liquid content, water, is enclosed inside of the cell through the process of endocytosis. And when it's water or any liquid content, we call it pinocytosis. And the pinocytosis vesicles. But what happens after this vesicle is engulfed? It usually goes and fuses with the lysosome. A lysosome is a very dynamic cell organelle. We'll study what lysosome is in the later part of this chapter, but it goes and fuses with lysosomes. But if in case it does not go and fuse with lysosome, this vesicle travels away from the lysosome to the other surface of the cell and eventually goes out of the cell. And this type of process is called transcystosis. So pinocytosis, transcytosis or cystosis, whatever you want to call it, uh, is basically accomplish bulk transfer of dissolved substances across the cell. Things go across the cell. They are transferred in the process of transcystosis. And we have the third type of endocytosis is receptor mediated endocytosis. Receptors for many substances such as low density lipoproteins and protein hormones are integral membrane proteins at the cell surfaces. We need to remember for what type of substances did receptor mediated endocytosis occur. So we saw that phagocytosis usually for the dead remains of any other organism, let's say bacteria, as we read. Pinocytosis is liquid material, water, which is cell drinking, and those vesicles have liquid contents. But when it comes to low density lipoproteins and protein hormones, we have a receptor mediated endocytosis. We read what are receptors. Receptors are some integral proteins which are responsible for cell addition, cell reconstruction, and response to protein hormones. Here are we talking about those protein hormones. Are integral membrane proteins, as I said, at the cell surface. High affinity binding of such ligands to their receptors cause their protein to aggregate in special membrane regions that they invaginate and pinch off internally as vesicles. So we have receptor proteins, protein hormone comes, gets aggregated to this special membrane protein and pinches off internally as vesicle again. The formation of fate of vesicle in receptor mediated endocytosis also often depends on specific peripheral protein on the cytoplasmic side of the membrane. So as I said, the fate, the luck of that protein that came, the protein hormone that came, depends on what type of protein, the plasma membrane protein it goes to. And if it's a receptor, the protein membrane, the protein hormone will get engulfed in the vesicle. The occupied cell surface receptors associated with these cytoplasmic proteins and begin to invaginate a coated pits. As coated pits, we need to know what are coated pits. The occupied cell surface receptors associated with this cytoplasmic protein and begin invagination as coated pits. So these type of locations where we have this type of protein receptors, the cell surface receptors that are associated with the cytoplasmic proteins, we call them coated pits. The electron dense coating on the cytoplasmic surface of such pits contains several polypeptides, the major ones being clathrine. What is clathrine? It is a polypeptide that is elect that acts as an electron dense coating on the coated pits. That's it. I think we should highlight clathrine. We need to know that clathrine is an electron dense coating on the coated pits. What is clathrine? A polypeptide, basically a protein. Clathrine molecule. This is the bad habit of this book. It just, in explaining one topic, it uses so many different words for some one specific thing. You just lose your mind sometimes reading. They had no need to call a protein a polypeptide in this sense, but they just said polypeptide, complicating things unnecessarily. Clathrine molecules interact like the struts of geodesic dome, forming that region of cell membrane into cage-like invagination that soon pinches off into cytoplasm as coated vesicle. So, cage-like invagination, the geodesic dome. This this is basically a term given to the vesicle that is coated with clathrine. So this type of clathrine coated pits is what responsible for invagination of this uh, protein hormone that came. There should be a figure 2.7 that explains this in better way. We need to go check table 2.7, figure 2.7. Yes, so this is receptor mediated endocytosis involves regulated membrane trafficking. So we have a clathrine coat here, guys. This so this pit which has a clathrine coat is called coated pit, and we have ligands, or you can also call them protein hormones, gets attached here, and then are termed into are closed into this type of vesicle structure. And any vesicle that is on the outside coated with clathrine coat is called coated vesicles. Let's read what happens to this coated vesicle with the receptor bound ligands inside. Okay, another type of receptor mediated endocytosis promotes in very thin cells produces invaginations called caveolae. So another type of receptor mediated endocytosis promote very thin cell produces invaginations. So another type is just this type of invaginations. The pit that was made, or you can call it an invagination, is called caveolae, that involves a family of internal membrane proteins called caveolins associated with diverse peripheral proteins called cavins. So we saw coated pits. These coated pits are coated with clathrine. In case of caveolae, these caveolae are uh, associated with the membrane proteins called caveolins, and the proteins uh, membrane proteins called caveolins associated with diverse peripheral proteins called cavins. We need to know what caveolae is. It's important. Caveolae, caveolins, and cavins. So the family of these proteins is called caveolins, and mostly this type of diverse peripheral protein is cavins. And when we have this coating to an invagination, we call it caveolae. In all these in all these endocytotic processes, the vesicles or vacuoles produced quickly enter the fuse with endosomal compartment, a dynamic collection in the peripheral cytoplasm of membranous tubules and vacuoles. The clathrine molecules separate from the coated vesicles and recycle back to the cell membrane of the formation of new coated pits. Give me a second, guys. I want to check if things are okay again, So, oh, do we have a viewer already? Oh, we have one watching. We have three people watching already. That's nice. Okay, so let's get back. What are endosomal compartments? It's a dynamic collection of peripheral cytoplasm plasm of membranous tubules and vacuoles. It's called endosomal compartment. Let's highlight this. So these vesicles that we just created, they just come and gets fused with this endosomal compartment. It is basically a dynamic collection of this type of vesicles, vacuoles. The clathrine molecule separate from the coated vesicles and recycle back to the cell membrane of the formation of new coated pits. Vesicle trafficking through the endosomal compartment is directed largely through peripheral membrane G protein called Rab protein, small GTPase that binds guanine nucleotides and associated protein. Important things, guys. So when they deploy this material to this dynamic collection called endosomal compartment, the clathrine protein travels back to the plasma membrane, creating the coat that will again be needed to create the coated vesicles. But vesicle trafficking that happens at this compartment uh, is directed largely through G protein called lab protein. So it is direct this compartment that we have of vesicles is directed to lab protein. It's a small GTPase. Whenever in biology we know that ASE, lactase, protease, GTPase, these are the molecules that cleave them. So small GTPase that bind guanine nucleotide and associated proteins, Rab protein. We need to know that vesicle trafficking through endosomal compartment is directly largely through the G proteins called lab proteins. They are G proteins. They are G proteins, basically GTPase, because they cleave guanine nucleotide. Okay, as shown in the figure 2.7, phagosomes and pinocytotic vesicles. We'll go to figure 2.7. Phagosomes and pinocytotic vesicles. Let's quickly review what happened so far. Receptor-mediated endocytosis. There is a cell membrane. We have multipass proteins that are integral proteins. Some of those are receptors. The role of receptors is cell adhesion, cell recognition, and also response to the protein hormones that come to. We have now a hormone coming to these receptor proteins. These receptor proteins basically create a pit ready for invagination of this hormone. This pit is coated, it's called coated pit because it has a coating of a protein. This protein is clathrine protein. This clathrine protein is responsible for this coated pit. We have a hormone and we engulf it and create a vesicle. This vesicle is called coated vesicle because it has a coating of this clathrine protein. This vesicle travels to a big pool of endosomal compartment where we have all the vesicles emptied, and this coated protein, the clathrine protein, travels back to the plasma membrane for creating new coated pits. Now we have this endosomal compartment where we have lot of this hormone material, etc. is directed largely through a protein called Rab protein. What is Rab protein? It is a GTPase. Basically, this protein cleaves the guanosine nucleotide. Let's see now what is important when it comes to Rab protein and what happens next. Phagosomes and pinocytotic vesicles typically fuse with lysosome within the endosomal compartment for digestion of their content. Simple. We phagosome some material or through phagocytosis going to digest it. We have pinocytosis done, cell drinking, we're going to digest it. through lysosomes. But what happens to this content of, let's say, hormones or ligands that we have? Let's take a look. While molecules entered by receptor mediated endocytosis may be directed down other pathways. What are these other pathways? The membranes of many late endosomes have ATP driven hydrogen H plus pumps that acidify their interiors, activating the hydrolytic enzymes of lysosomes, and in other endosomes, causing ligands to uncouple from their receptors, after which the two molecules are stored into separate endosomes. The receptors may be sorted into recycling endosomes and returned to the cell surface for refuse. Low density lipoprotein receptors, for example, are recycled several times within cells. Other endosomes may release their entire content at a separate domain of the cell membrane, transcytosis, which may occur in many epithelial cells. So now these contents goes to different pathways. What are these pathways?